here to about here. Okay? Bless you. Then the ciliary body goes from about here to here. Okay? And then the most anterior portion from here to here is the iris. Okay? So again, choroid, and then here to here is the ciliary body, bless you, and then here to here is the iris. Okay? The choroid, its main function is that it supplies blood to the wall of the eyeball. Okay? There's a lot of blood vessels and stuff in it. The ciliary body, so that's this portion from here to here, it has a lot of smooth muscle in it, and what happens is there are little strings, which they do call ligaments, although technically we say ligaments connects what? Bone to bone, right? So this, of course, would be the exception to that rule, okay, where these little strings are called ligaments as well. And they, of course, connect the ciliary body to the lens. Okay, so they help anchor the lens and hold the lens in place. Okay? And then finally, the iris is this portion here, and that's the colored part of your eye. Okay, so if someone has hazel eyes, blue eyes, brown eyes, the colored pigment is in the iris area. Okay, and of course, there's an opening in the middle of the iris. Of course, that's the pupil. Okay. Now, the iris itself, not only does it have a lot of colored pigment, gives your eye its color, but it's got a lot of smooth muscle in it, okay? And remember we talked about the sympathetic and parasympathetic nervous system, right? The sympathetic nervous system can actually send nerves to the iris and cause certain muscles to contract, which does what to the pupil? Dilate. So if you look at this picture here, this is the normal pupil, let's say, okay? And you can see there's the blue is of course the iris, right? So you can see the smooth muscles in the iris. Some of it is a circular muscle around it, and then you've got these more straight muscles out here. Those are both smooth muscles, okay? So of course when the sympathetic nervous system kicks in, so like I said, someone points a gun at your head, a lot of sympathetic activity goes out, goes through nerves, and goes to the iris, and it causes which muscles to contract? The circular one or these more linear ones? The linear ones, right? Because you can imagine if the linear ones shorten, then the pupil gets larger, right? Of course, the parasympathetic would be the opposite, right? So if you send out a lot of parasympathetic signals, and of course it would go out through what nerves? Which?
Ocular.
Right. Okay. You just need to know it would be one of which ones. 3, 7, or 9, right? Remember, there's a smooth muscle in the head and neck area, right? Parasympathetic nervous system can innervate any smooth muscle in the head and neck area through one of those nerves, 3, 7, or 9. It's really 3, but you don't need to know the details of that, okay? Just need to know it's one of those 3, okay? So, of course, the first neuron in the parasympathetic nervous system that would send the messages to here is located where? Brainstems, okay? So that nerve, in this case the oculomotor nerve, will send fibers or its axons to innervate which muscles? The circular or the linear ones? For parasympathetic. Circular ones, right? So when this muscle contracts, what's the pupil do? Constricts, right? Just like the smooth muscle in the wall of the blood vessel, right? We said when that muscle contracts, the blood vessel does what? Constricts, right? So same thing with this muscle, right? If it contracts, it constricts the pupil, makes it smaller. Okay? All right, so we've talked about the fibrous tunic, which we said in the back was what? Sclera, and in the front it's cornea. And we talked deep to that is the vascular tunic. We said the structure in the back of the vascular tunic is what? Choroid, the middle one here. Ciliary body, and then anteriorly iris, right? And of course the hole in the middle of the iris is the pupil. So remember, the pupil the size of the pupil is dictated by the iris, right? Okay. So what structure controls the amount of light that enters the eye? What controls the amount of light that can enter in here? Iris, right? Because light has to enter the eye through the pupil, right? And of course, what controls the size of the pupil is the iris, right? So if your pupil is very dilated, of course, a lot more light can get into your eye. If your pupil is very constricted, very little light is in your eye. So if you notice, if you walk into a very bright room, what does your pupil do? It constricts, right? Because there's too much light, so it wants to lessen the amount of light that gets into your eye. If you go into a dark room, what does your eye dilates, right? Because you want to try to get more light into your eye. So of course, the iris, of course, controls the size of the pupil, which in turn controls the amount of light that gets into the eyeball. Make sense? Okay. All right. So we've talked about the first two layers. Now let's talk about the deepest layer. That's the yellow one here. Okay. Goes from here to here. This is known as our sensory tunic. It's a sensory tunic because this is where the sense of where you receive the light information, the receptors for that sense. Talk about the five senses, right? The sense of vision here. The receptors for the sense of vision is located in this layer. That's why it's a sensory tunic, okay? And that is primarily made up of the retina, okay? So all this yellow stuff here is the retina. So the retina contains the photoreceptors, and those are, again, the receptors that capture and the light energy that enters your eye, okay? What are the two photoreceptors in our eye called? Anybody know? Rods and cones, okay? So the rods and cones, as we'll talk about in a little bit, the rods and cones, as you can see, responds to more dim light, whereas cones respond more to bright light, okay? And the rods, so basically the rods are involved in more your peripheral vision, whereas the cones are responding more to the specific object that you're focusing on, okay? So if you're looking at a specific object, the visual information from that object is actually being received by the cones. If it's just your general peripheral vision, it's receiving, the rods are the ones that receive that information, okay? So, there's an area on the retina called the fovea centralis, and this is where the greatest concentration of cones are located, okay? So when you're staring at an object and you're getting visual information of a specific object, the light rays that come off that object should be focused onto the fovea centralis, because that's where the greatest concentration of cones are. So these are the photoreceptors that receive very detailed light information, okay? Whereas the rods, again, these don't receive detailed light information. It's more general background information, okay? So let's go into the retina a little bit more detail here, okay? So remember, the retina was on that picture of the eyeball we just showed was that yellow layer, right? Okay. So if you look at it up close, the retina is like from here to here, okay? So this is all within the retina, that yellow layer there. So what happens is, okay, this is the back of the eyeball, so that means the front of the eyeball is here. So as light comes in this direction, so it'll be coming in this direction, light's gonna go through, and then the rods and cones are these guys, the purple and yellow ones, okay? They're gonna receive that light waves, okay? So what they do is once they receive that light information, they're gonna send that information through impulses or depolarizations, action potentials, right? They're gonna go through its axons and they're gonna send it to these cells, which then send them to these cells. These cells are called ganglion cells, and you can see how their axons come out of their cell bodies. And those axons, actually all from those ganglion cells, all come out of a nerve here, right? And what nerve is this? Optic nerve, right? Okay? So the optic nerve is made up of axons, that come from these ganglion cells, okay, that are in the retina. So of course the ganglion cells are receiving that light information that are originally captured by the rods and cones. So what you guys need to know are the rods and cones are the photoreceptors. They initially receive the light information. They eventually send that information to the ganglion cells. And then the ganglion cell sends that information through impulses or action potentials through its axons, right? And those axons are going to go out the eye through the optic nerve. And then from there, it's going to go to where? Well, yeah, then to the optic chiasm, from there those fibers go to which neuron cell bodies next? Thalamus, right? Because all sensory input pretty much has to go through the thalamus first, right? And then the neuron from the thalamus sends that information to where? The visual cortex, where the cells there interpret the visual information, right? And of course, the visual cortex is located where? Occipital lobe of the brain, right? Okay, does that make sense? All right. So the rods and cones again, well, we'll talk more about that in a little bit. So remember we said the blood supply to the wall of the eyeball comes from the choroid, right? However, if you look into an eye, you'll see there are blood vessels that go in and out of the eye at the area where the optic nerve starts too, okay? And that's, you have, these are your retinal artery and veins, but so that also supplies blood to the wall of the eye as well, okay? So it's not just the choroid, but you do have other arteries that supply blood to the wall of the eyeball, okay? So again, this spot here in the eyeball where the optic nerve starts, and then where the blood vessel, the artery goes in, brings blood in, and the vein brings blood out, okay, all comes out of this spot called the optic disc, okay? So if you look in somebody's eyeball, okay, most of you, I'm sure have gone to the doctor's office or gone into the, if you go to a doctor's office, they hold a little small ophthalmoscope and look in your eye, or if you go to an eye doctor, they have you go into a slit lamp, you put your head on that thing and they bring this thing really up close to you, right? And they look in your eye. Well, they're looking into your eye, they dilate, oftentimes they'll dilate your pupil, right? So it makes it easier to see into your eye. And of course, what they're looking is in the back here, this is what they should see, okay? So, what is this? That's the optic disc, okay? So that is this. Remember, there's artery that comes in and a vein that goes out from there, right? So you can see the artery that comes in and how it branches. Okay? So that's the optic disc. This is all part of the retina. Okay? So, by looking into your eye, you can actually visualize, you can actually see a lot of different types of medical disease processes that can affect the retina. Okay? You can see what on this picture, these blood vessels look nice and clean. You got a nice round, sharp-looking optic disc. In this picture, you can see the blood vessels. You've got all these white spots around it. You've got some black and white spots on the retina. These are examples of disease processes. So people who have high blood pressure or diabetes, they can start damaging their small vessels, their arteries. They can start looking like this. So you can actually visualize disease of your blood vessels by looking at somebody's eye who has bad diabetes or high blood pressure. Sometimes you can have infections that affect the eye, such as this one. And then in this one, what can happen is, I know it's hard to see, but bless you, imagine this is a membrane flap that's coming down and actually should be stuck up against the wall, and then it's kind of a flap that's come down. So it's your retina that's coming, what's that called? Retinal detachment, okay? So, so obviously you can see a lot of different things and a lot of pathology in people's eyes sometimes. Okay. So let's talk about the internal chain.